I chose the titles Three Spikes and You're Out because there is, of course, a big discussion right now. How many vaccine shots do we need? And alternative title would have been a healthcare worker study and its consequences. And actually, there was a close collaboration with different partners here in Munich. So everything started when we had a very severe first wave in Munich, much more severe than in the rest of Germany, because we were just so close to Italy and to Austria, where all these pandemics in Europe at least started. And what we showed here is the COVID-19 cases we had already early 2020. And what we also showed here is the positive, PCR positive staff. And what we actually started this study because everybody after the first wave thought, well, I've got it. I had the infection and I don't have to care anymore. But obviously it was not the case. But that's basically why we started a seroprevalence study. And also this whole study is now in the meantime published, showing that basically with appropriate hygiene measures, you can really prevent infection of staff because we immediately introduced that. And you can see the peak of hospital worker infection was much before we had the peak of patients. So the measures really work. What we found in this study, including 4,554 healthcare workers from our university hospital, rechts der ISA here, is that 108 of those had been infected already during the first wave, giving a seroprevalence of 2.4%, which for Germany was relatively high at that time point. And that gave us the opportunity to sort of study this group over time. And what we did is we studied, obviously, we first identified them, as you can see here on the left-hand side. That's the anti-N antibodies, so really indicating the infection, not the surface antibodies. And then we studied their T-cell responses, and we found that those who tested seropositive, they also had a T-cell response in different gamma and IL-2 response against the S1 and the S2 spike protein, whereas the naive ones, where we could not measure any antibody, they had very low, if any, T-cell responses. So there was a clear difference. And what we also did is we studied the neutralizing antibody response using a real virus neutralization assay, which we had built up at that time in the BSL-3 lab. And almost everybody 2 months after the first challenge after the infection had neutralizing antibodies. However, if we looked at that again 3 months later, so 5 months after the probable infection time point, antibody titers and particularly neutralizing antibody titers had already dropped. Not in everybody, there were a few individuals where they were rather stable, but in general, in a lot of them, neutralizing antibody titers had dropped. And in a significant proportion, they were below the level of quantification or even below the level of detection in our assay, which is relatively sensitive. So there was at that time point a 2.4% seropositivity in our Munich University Hospital, which is, I think, very representative in healthcare workers after the first wave. And we saw this rapid decrease of neutralizing antibodies after SARS-CoV-2. Then we kept on monitoring our cohort, and then the vaccine came. This was then obviously December 2020, so we already had a 9-month observation time of our convalescents and also a group of naive individuals, which we matched concerning sex, age, and also working place, because we wanted to have them at a very comparable risk. And then everybody was vaccinated. The convalescents were vaccinated, and the naive ones were vaccinated. And as everybody knows at that time point, they got 2 doses of Comirnaty, so the BioNTech mRNA vaccine, 3 weeks apart. And what we did is after the first vaccination, we called everybody in and asked them to donate blood. This was then a group of 91 convalescents and 53 matched naive individuals, which agreed to participate also in the vaccine cohort, which we then called VACCOMRI, after CCOMRI, which was more on security, and this was more on vaccine responses. And we did the same 2 weeks after the boost vaccination, and we studied their antibody responses, we studied their T-cell responses, and of course, we also checked whether anybody had been infected in the meantime. But there were very few which dropped out due to that. And then we said, okay, we want to follow them up, and we did so and had visits again in May, which was after 3 to 4 months, in September, which was after 7 months, and then there was the third vaccine dose coming up, and almost all of our participants got a booster and still continue to participate in the study. So we had a very nice cohort with a really long-term follow-up. Now that's basically now already showing how it goes into 2020 and 2022 and 2023. So more the future, how we plan it, but of course we want to continue following up. The data which we obtained on the dynamics of spike and nucleic capsid specific immunity during this long-term follow-up before and after vaccination, they have been summarized in a paper which recently came out in Nature Communications, and they will not be the focus of my talk here. So if you're interested in that, please go and look in that paper. I just want to show two figures, or one figure out of that paper. The one which I think is very instructive already here. So what we did is we had a surrogate neutralization assay and determined surrogate neutralization activity in the blood of our participants. And the convalescents already after a first vaccine shot had very high titers, whereas the naive participants had low titers. And only after a second vaccinations, the naive ones picked up and got at the, or came up at the level where the convalescents already were. And that then basically was just one of the data sets. There were many international data sets on that, which indicated that a very short booster, this was 3 weeks in between, if you had an infection before, does not really help you. And one vaccine shot is sufficient if you had an infection before, already indicating that the infection can really substitute for one vaccine shot. And it looks also very similar when you look at T-cell responses, which we, by the way, here do with a multicolor fluorospot assay, which has the advantage that it's relatively easy to standardize over such a large cohort. And what it does, it can measure different cytokines, up to 6 different ones. And then you have multicolor spots coming up if you have dual or triple or quadruple positive T cells. However, the 2 dominant cytokines we found were always interferon gamma and IL-2. So what happened was that this is again the convalescents here at a relatively late time point. In the convalescents, we could boost the T-cell response by one vaccination. A second one did not help anything. But in the naives, obviously, the second booster also increases your quantitatively increases your functional T-cell responses. Now, again, we followed them up after vaccination. And what we found is that over time, your neutralizing antibodies, they drop again. And that is true in the convalescent group as well as in the naive group. In both groups, the antibody levels dropped significantly. So both responded, but the antibody titers dropped. Now we said, okay, it would be very interesting to really be able to show or to see what the neutralization capacity of these sera would be. And we also wanted not to only test against the original Wuhan, or we had an isolate which in the lab, which we isolated ourselves. So it's an EU1 strain, but still the original D614G variant of the SARS coronavirus. And we wanted not only to test the neutralization activity against this variant, but also against all the variants of concern which had appeared in the meantime. This was alpha, beta, gamma, and delta at the time where we started to do that. And we could only do that because Oliver Kepler's group at the LMU had established a high throughput cell culture neutralization assay, where basically you use the primary SARS coronavirus isolate, which you expand and titrate in cell culture. Then you use your donor serum species, dilute that in cell culture medium. This can be done automatically with a robot. Then you incubate the serum and the virus, and you bring it onto cells. And this is a cell line which is an immortalized cell line which is stably transduced with the receptor for SARS coronavirus, so with ACE2. And on this cell line, the virus elicits a cytopathic effect. And that is basically what you then can measure in really larger scale or in high throughput. So simply the cytotoxicity with a cell titer glow assay. And that can be scaled up to a 384 well format and allowed us to test 100 samples per time point, a total of 476 serum samples for the original strain, but also all the variants. And what we found doing that is that in general, you can already estimate that here, but see that better in the next slide, there is a little higher neutralization capacity of convalescents compared to naive. This is here against the alpha, the beta, the gamma, the delta, and the Omicron VOC. But what you also see is that the beta VOC already has some sort of an immune escape. So you need higher titers of serum to neutralize the variant. But Omicron has a huge capacity to escape neutralizing antibodies in our participants or volunteers, sera. And this was not that obvious directly after vaccination, but if you wait longer after vaccination, so that's the value 2 weeks after vaccination, and that's the value 4 months after vaccination, then you see really the ability to neutralize Omicron compared to the EU1 strain, that's what we show here, that drops significantly. So Omicron is really able to escape serum neutralization. Now, when we look at that over time and really look at the different variants, and let me guide you through this a little bit complicated figure here, then we first started off 8 to 9 months after infection in the first wave, as I described, and we compared convalescents compared to naives. And again, the convalescents, of course, they had a contact, they had neutralization capacity, and that was still there 8 to 9 months after their infection. However, for Omicron, it was almost gone. So it's always hard to say where the cutoff of a neutralization capacity is. Here we said it's 1 to 10, and there is quite a number already below that threshold. And you also see that there is obviously a difference to the other VOCs. Now, what does it look like 2 weeks after one vaccination? So what we've seen before is that the capacity to neutralize virus really jumps up in convalescents with one vaccination. And that is then relatively high for alpha, beta, gamma, delta, and the original virus. And again, is lower against Omicron. But after one vaccination, already our convalescents were quite well able to neutralize also Omicron, whereas the naives were not. The naives really needed 2 vaccine shots, and that's what's shown here on the right. That's 2 weeks after the second vaccination. There you see that also the naives develop the capacity to neutralize the variants and also some capacity to neutralize Omicron. However, that's really low. And it also indicates that the convalescents always come out a little better after only 1 vaccination than the naives do. Seeing that, we wondered, well, is that now really the quantity of antibodies, or is there maybe the quality of the antibodies different between those 2 groups? And with that question, we then went on. But maybe first, let me show you the data, what happens after a 3rd vaccine shot, so after a booster shot. And here, that's now the time row, basically what you've already seen. After infection, after 1 vaccination, after 2 vaccinations, 4 months after the second vaccination, 7 months after the second vaccination. And then here comes the booster shot. So even in the convalescence, there is some sort of a booster. However, the booster is really important, and that's the neutralization capacity against the Delta variant. The booster shot is really important for those who had not have an infection before, so for the naive ones, because they were relatively moderate in their capacity to neutralize even the Delta variant. However, if they got a 3rd vaccine shot, that capacity really jumped up. And that holds also true for Omicron. In Omicron, it becomes a little more obvious for our convalescents. So after 1 vaccination, a little drop over time, then after a 2nd vaccination with the distance, so at the same time where the naives got their vaccination number 3, they had a high capacity to even neutralize Omicron, and also the naives who were very low after their 2 vaccination, and that exactly fits the clinical observation that 2 vaccinations are just not sufficient to neutralize Omicron. If you have a 3rd vaccine shot, then your neutralization capacity really increases. Now the question comes up, is that the quantity or is that the quality of antibodies? So we first looked at antibody titers, so the quantity of antibodies, giving here in this international BAU, so binding arbitrary units relative to the WHO standard. And what we found is that looking at the convalescents, there was high titers after 1 vaccination already. It dropped over time. And again, a high titer when they got their 2nd vaccination at the same time point where the booster, the 3rd shots were given. It looks a little bit different if you look for the naives. The naives, they go up rapidly after the 2nd vaccine shot. Then they drop quite a bit, and then they jump up again after the vaccination number 3. And that does not 100% correlate to what you've seen on the slide before, and that's why I show it again here. So the neutralization capacity against Delta, because although the convalescents dropped in their titers significantly, that's a logarithmic scale, so a factor of more than 10, their neutralization capacity was relatively stable. And also the drop in the quantity of antibodies in our naives was not reflected at the same level in the neutralization capacity. So that really was a little different. So we thought, okay, it could either be the breadth of antibody responses, which is certainly higher in convalescents than it is in vaccinees, but it could also be the avidity. And we looked at the avidity of the serum antibodies of our 2 groups. And there we really saw that the avidity correlates very, very well with the neutralization capacity. So our convalescents, which remained relatively stable over time, it took a little bit, but then after 1 vaccine shot, they had developed a high avidity, and that also remained very stable. And the same was true for the naive ones. They take a little time to develop a higher avidity. So there is an increase. There is a drop in antibody levels from month 4 to month 7. However, there is an increase in avidity of the antibodies between month 4 and month 7, reflecting the maturation of the B-cell response. And that then again increases after a third vaccine shot, so after a third antigen exposure. And then, of course, we asked ourselves, well, does that necessarily have to be 3 vaccinations? Or would it maybe also fine if you have a breakthrough infection? And what we did here is we added to basically the data you've already seen, and that's the naives 2 weeks after the second vaccination, 2 weeks after the third vaccination, the convalescents the 2 weeks after the last vaccination. And then we added 2 groups which we had obtained in a different study, in a vaccine breakthrough study. And we had breakthroughs against or happening with Delta, and we had breakthroughs happening with Omicron. And what we found is that the breakthrough happening with Delta or Omicron brings you from that level up to almost the same level as you have if you have a third vaccine shot. And that holds true for, again, all the variants, the Alpha, the Beta, the Gamma, the Delta, and even the Omicron variant. Interestingly, the Delta breakthrough is a little less potent, or seems a little less potent than the Omicron breakthrough is. However, one has to say this is a relatively short time after the breakthrough infections, only around 10 days after breakthrough infections. So probably these values will still increase. And that then, when the data appeared, made Eric Topol to say, well, 3 spikes and you're out. And that's also why I chose that as a title for my talk. Now, very last slide, asking the question, does that now again correlate with avidity or antibody titers? And the answer is it does correlate with avidity of the antibodies. So you clearly see that the third vaccination, or the booster vaccination, but also the breakthrough infection really increases the avidity significantly. And that is probably why you then have a much better neutralization capacity. And when you look at that over time, that's Delta breakthroughs where we had the opportunity to study them after 1 week, 2 weeks, 3 weeks, 4 weeks. Then you see it takes a little bit after your breakthrough infection, but after 2 to 3 weeks, you're really at high levels of avidity of the antibodies. So again, it's the quality, which is very important, the quality of the antibody response, maybe even more important than the quantity. And with that, I'm at the end. I'd like to thank everybody who contributed to this study, which is the people in my lab, also in the diagnostics here, the people in Oliver Kepler's lab, establishing and also performing the high throughput assay, Percy Knolle's group, who organized all the clinical trial, and the Helmholtz group, which did all the immune monitoring. And I thank you for your attention. And of course, also all the study participants who really followed us up for now almost 2 years, always come back, donate their blood. And that's, I think, worth quite a big thank you.
Yeah, thank you very much for this exciting talk. We're a little bit over time already. I don't know whether you heard my warning, maybe not. But I think we still can have 1 or 2 quick questions, quick answers. If I have the first one, if I understand it right, then our, let's say, vaccination policy at the moment is fully supported by your data. Is that correct?
Yes. Now we have a new recommendation that persons at risk should get a 4th vaccine shot. That's based on the Israel data. However, I think everybody looking at that study recognizes that the gain which you have by a 4th vaccine shot is not that huge. But for risk groups, I think it's a good decision to offer that.
Yeah. So are there any further questions? Please put a question mark in the chat. There's a question, I possibly read it for time reasons, from Dirk Görlich. how cross-reacting T cell response is there with other coronaviruses?
Yeah, that's a very good question. There is cross-reactive T cells, which you can find. However, we selected our peptide pool for non-cross-reactivity, and we're currently studying whether it's cross-reactive also against the Omicron variant. The epitopes are rather well preserved. One has to say there is also studies out by Alessandro Sette's group in the meantime. Our own data are not ready yet.
This next one is from Andreas Radbruch.
No, I just wanted to point my attention to Michael Reith and Klaus Rajewski.
And Klaus Rajewski. Probably these two can still ask their questions themselves, and then we have to carry on, I would say.
I just wanted to ask what avidity exactly means. When you talk about antibody avidity, what is this, an affinity measurement, or what kind of measurement is this?
We call it avidity because basically what we do is we test the binding strength under high salt conditions and quantify antibody binding without a high salt exposure and with a high salt exposure. And then we see how many antibodies survive that high salt exposure. And that's the relative avidity which we gave.
Thank you.
Michael, please.
Yeah, okay. What we also found it actually, when you looked at your early group of infected people, then you found that many of them had actually no antibodies, or rather few antibodies. And they made a booster response after the first immunization. So how do you see this? This is actually a puzzle for us, that several of your first group of individuals in the early times of the infection, they had hardly any antibodies, but they made immediately a booster response. So that is for me a puzzle of this pandemic, which you also noticed. Do you have any ideas on this?
Yeah, I mean, what we see that is they already have a T cell response, and that's mainly a CD4 T cell response. And my take would be they certainly also have a memory B cell response, which they had the chance to develop over time. And again, we saw that the antibody titers dropped very much, but the neutralization capacity did not drop that much. So it's not only the titer, again, it's a mixture of the titer and the quality of the antibodies.
Yeah, I think that the virus somehow, you get the memory, but you don't get the plasma cell development very well.
Yes, that's what I think.